Hello, my name is Bridley Jenkins, and my disease is botulism, and I am going to be discussing the acetylcholine signaling pathway. So first, I'd like to go ahead and address the concept map that I have done, and I chose cell-to-cell signaling systems, that type of cell communication. And there are two types, local and distant. Local being paracrine signaling and distant being endocrine signaling. And paracrine is local because we have communication with a nearby target cell in which the original cell is creating its own molecules called those ligands. And once those ligands are released from that original cell, if the target protein has a receptor for that specific ligand, then that ligand can then bind to that cell and then elicit a response. And with endocrine signaling, instead of our ligand binding to a nearby target cell, we actually have it traveling all the way to blood vessels and entering the bloodstream to flow to its proper destination to get to whatever body cell needs it. And with these responses, we have four ways in which the signaling continues. So one of those ways is the protein kinases, and these are enzymes that actually transfer a phosphate group from one to another, which helps activate the other protein kinases and keep the cascade going. And then we have cyclic AMP, which acts as a second messenger, and it binds to cyclic AMP dependent kinases, and it also helps those kinases to phosphorylate in order to elicit a continued cell response and reaction. And we also have calcium ions acting as second messengers, and they're activated by those ligand gated ion channels. When a ligand binds to the ion channel, it's opening and it's able to trigger change in protein shape and charge. And lastly, we have scaffold proteins, and these are serving as docking stations for multiple protein partners in the cascade so that they can be closer to each other, and this helps the message between the cell membrane and the nucleus to happen a lot faster. So next I'm going to go ahead and talk about botulism. So according to the CDC, botulism is a rare but serious illness caused by a toxin that attacks the body's nerves and causes difficulty breathing, muscle paralysis, and even death. And this toxin is caused from the Clostridium botulinum bacteria. And this bacteria can thrive in environments with little oxygen. It can thrive in the infection of a cut producing that toxin, and it can also thrive in a baby's intestinal tract when those bacterial spores are growing. And those are three different types of botulism. And with our symptoms of botulism, we have double or blurred vision, drooping eyelids, slurred speech, difficulty swallowing or breathing, thick feeling tongue, dry mouth, and muscle weakness. And specifically with infant botulism, which is the most common type, we have the baby appearing lethargic, it feeding poorly, constipation, a weak cry, and poor muscle tone, so the baby looking kind of floppy. And the treatment option right now for botulism is the drug called antitoxin, and antitoxin is actually preventing the botulinum toxin from causing any more harm to the body. However, it cannot reverse any of the damage that the toxin has already done. So, for example, if the toxin paralyzes your breathing muscles, your doctor may put you on a ventilator until you can breathe on your own, because although the antitoxin was given to you, it was able to stop any more of the paralysis happening, but it could not reverse any of the paralysis that has already been done to your breathing muscles. So, with the acetylcholine pathway that we have, the normal one, we have two types of receptor proteins. We have the nicotinic acetylcholine receptor, which is a ligand-gated ion channel, and we have the muscarinic acetylcholine receptor, which is a G protein coupled membrane. And with the binding sites, we have two binding sites for the nicotinic receptors and one binding site for the muscarinic receptors. And all of these binding sites are extracellular. Now, the characteristics of the signaling pathway with a ligand, both the nicotinic and muscarinic receptors have acetylcholine as their ligand. We have two second messengers being acetyl-CoA and choline. And our scaffold proteins, we have two of those being SAP kinase substrate 1 and rhabdin. Now, with protein kinases, we also have two of those, being protein kinase T and calmodulin-dependent protein kinase 2. And their functions are to phosphorylate choline acetyltransferase onto the amino acids serine 440 and 39456. Now, choline acetyltransferase is an enzyme that's responsible for catalyzing and regulating acetylcholine synthesis, so it's very important in this acetylcholine pathway that it gets phosphorylated so it can continue to be created and help this process move along. Now, we have three different G proteins. There's G alpha O, G alpha Q, and G alpha S, which are these G proteins within the G alpha subunits. So now we're going to discuss how the botulism toxin works. So the first step is that the free end of any of the 8 botulinum neurotoxins binds to the cholinergic receptors, so the nicotinic and muscarinic receptors we talked about earlier. And with that, the botulinum toxin then passes through the cell membrane and the cytoplasm via that receptor-mediated endocytosis, so that vesicle being created for it to be able to enter into the cytoplasm. And then the botulism toxin is able to cleave to the snare proteins, and our snare proteins are acting as our relay proteins. And I must mention that the botulinum toxin is acting as our ligand. And it's also very important to mention that the scaffold proteins called septins contribute to the intracellular stability of the light chain of the botulinum toxin and keep the light chain from undergoing intracellular degradation. Hence, making sure that the toxin can actually cleave to the snares, because if it's degraded, it wouldn't be able to do exactly that. So this keeps it stable and able to cleave to those snares. And there are also downstream receptors that are non-functioning within this pathway. Next, we have the toxins cleaving to those snares, and that cleaving actually prevents the snares from forming a synaptic fusion complex. And without this synaptic fusion complex, a synaptic vesicle cannot fuse with the nerve terminal membranes. And as a result, no acetylcholine is released, therefore causing the muscle paralysis and the botulism that we see. So here is a drawing I did of the normal cell pathway. We have the synaptic vesicle, and we have our snare proteins creating a synaptic fusion complex with that. We then are able to have our vesicle and terminal membranes being able to fuse together, and then that inhibits the release, or not inhibits, but creates the release of the acetylcholine, and they're able to attach to the acetylcholine receptors, and therefore the muscle fiber is able to contract. Now, we have our abnormal cell signaling pathway drawn here. So first of all, the botulinum toxin, as we talked about, is endocytosed into the cell membrane, into the cytosol. And we have our light chain of the toxin actually cleaving to specific snares, and that causes us to not be able to have a snare complex forming, and as a result, our membranes do not fuse, and so no neurotransmitter is released, no acetylcholine is released, and then our muscle fiber is then paralyzed. Now, with the cell response and signal termination, so a cytoplasmic cell response is generated, and this is due to the toxin binding to nerve terminals and translocating into the neuronal cytosol. Now, the signal is terminated when the intrinsic GT-PACE activity of G-alpha protein leads to the hydrolysis of GTP to GDP and the reassociation of the G-alpha GDP in G-beta-Y subunits. So this termination really all has to do with GTP, hydrolysis to GDP, and that reassociation of those two subunits. here are my resources that I used. I used quite a few. I used YouTube, the National Institute of Health. I used the CDC, the Mayo Clinic, ProQuest, and a lot of other scientific research websites. So thank you for listening to my presentation, and I hope you have a wonderful day.